Hey everyone, welcome to today's episode of the Biometrics Leadership Lab. We are here to support you on your biometrics journey, giving you the tools and knowledge you need to grow from lead to leader. Today, I'm wrapping up my 20 Things series with 20 things your statistician wants you to know. And I'm thrilled to bring this great panel today, which covers sponsor, CRO, and consulting aspects. Thank you for joining us live. And if you're watching a replay, thank you for taking the time to catch up. You can ask questions during the event on the notice on LinkedIn, and we will see them. However, we can't respond in writing. So if we don't answer your question live, look for an answer after the event. Before we get started, I'm going to introduce myself and the panelists. I'm Amanda Cross, founder of Crossfield Coaching and Consulting, an executive biometrics consulting agency dedicated to helping small to mid-sized companies and teams deliver biometrics in a better way. With decades of experience in clinical research, from statistician to chief operating officer, I leverage my cross-functional experience and training to help teams integrate, communicate, and collaborate to improve their service delivery. I also help functional leaders professionally develop themselves and their teams. And off to the panel, Elizabeth.
Hi, everyone. My name is Elizabeth McIlvaine. I am a doctorate statistician. I've been in the industry for 10 years. I am the founder and solo statistician currently at Aurelian Biostatistics, which is my consulting firm. Thanks so much for the introduction, Amanda.
Thank you. Adam J.
Hi, Adam Jacobs. I'm the head of biostatistics at Ergomed, a CRO. I'm based in the UK. I have always worked on the CRO side, and I've been in clinical trials since the 1990s. So, yeah, been around for a few years.
Perfect. And yeah, that's right. You get 2 Adams for the price of 1 today. So, Adam Ham, if you want to introduce yourself.
Hi, Adam Ham. I'm the Senior Vice President of Biostatistics and Strategic Consulting here at SDC. I've been in the industry going on 22 years now. Started in the early 2000s, and I'm very excited to be here with you all today.
Great. Before we jump into the 20 things, I always like to start with this question. So I'm going to ask each of you to give me about 1 sentence about what you think statisticians do in clinical research anyway. Who's going first?
I'll go first. Statisticians in clinical research. We help researchers understand how to collect data, how to analyze it, and how to interpret those findings.
So I will say that clinical research is a real team effort, and the statistician is an integral member of that team. We can't do it by ourselves, but the rest of the team need us to do all the things that Elizabeth just talked about. We need to think about study design and data and all those sorts of things that we do. But the important thing is we have to work collaboratively with the medics and everybody else in the clinical research team.
Yes, I agree on all points. I also have been on the CRO side all of my career. And in addition to all of that, I mean, you need to be part of the study design team. But as client facing, you also need to help clients understand how is what you are doing efficient? How will it save them time and money? And so there are a lot of things outside of the normal statistics that I get to do and that I've been fortunate to do over a long period of time.
Great. Thank you. We're going to thing number 1, and we're going to work to get through all 20 today. First, involve the statistician from the beginning of your program development. And I just want to make a note so everybody is aware and we're on the same page that the program is the entire planned clinical development program for a compound, as opposed to any single individual study protocol. And statisticians have a lot to say at the beginning of program development.
So if we're going to get through 20 things, I'm going to keep all my answers relatively short. I think as statisticians at the beginning of a program, we're responsible for a through line that is relevant to our function, which includes things like collecting the same data, collecting data the same way, refining hypotheses, and having that statistical voice to let you know how one study will inform the next as you move through the entirety of the program, and to also have kind of an eagle-eye view of the entire process from start to finish to market.
And I think the statistician is well placed to say, not only what data do we need to collect, but when do we need to collect it. So, obviously we want to have good evidence on efficacy, but we're not going to get to that until we've demonstrated safety in our phase 1 studies as well. So having that overview of the whole thing is something where statisticians can add value. And at the risk of repeating myself again, as one part of a multidisciplinary team, we need to work nicely with everyone else to do a good job of that.
Yeah, and what I'll add to that is that as technology evolves and machine learning and such, there are so many great tools out there right now that help you think through the program. Now, the statistician certainly has to give the right inputs, but I believe that with the advent of the technology and program development, it's becoming a lot more innovative.
So let's move on to the next step. Not only should you involve them at the beginning of your program development, be sure and involve them at the beginning of your protocol development.
I love this one. There is no fix for a poorly designed study. There is no statistical fix for collecting data wrong, messily, for collecting the wrong data. We are not magicians. We cannot fix issues that are inherent to the study that we're coming in on. Bring us in early so that we can get, our statistical knowledge into every aspect of the study. It is it's incredibly key to have a statistician help you design at the very least, and we really need to be involved throughout study startup, CRF building, EDC building, like through speccing out analysis data sets. We really need to be a part of the whole process, and it will save time and money down the road to have us be a part of it so that, so the problems don't crop up when you're 2 weeks from your database lock.
Yeah, and I just want to reiterate the point that a statistician cannot fix a bad study after the event. You need to have that study designed right in the first place. And the statistician's not the only person who's going to help you design it right, but that's certainly an essential part of the picture. And we need to make sure we've got a design appropriate for the phase of development. Are we trying to do a confirmatory phase 3 study and make sure we've got all our statistical ducks in a row, or are we doing something more early phase and exploratory and can maybe take a few liberties and perhaps have some fancy adaptive design features or whatever? But these are things a statistician can think about and come up with good suggestions for.
And speaking of adaptive studies, an adaptive study is not a fluid protocol that can change on the wind. So I would like to ask one of you to define what an adaptive study really is, and then give your comments about what it means in reality.
Yeah, well, I mean, adaptive, it can take on a lot of different shapes and sizes. And I get a number of clients that bring to me what they call an adaptive design. And from my perspective, it's a simple group sequential where you have hard stopping rules and such. And that is adaptive because you could make changes to the study in the middle of the study. There are also adaptive studies such as dose pruning, early phase oncology and such, where you are making decisions based on the accumulating data. And those could involve Bayesian methods, standard frequentist methods and such. But it is very interesting how non-statisticians define adaptive studies.
I mean, I think adaptive can cover a whole range of options, as you say, a simple group sequential design or a sample size re-estimation. Incidentally, almost every phase 3 study should have a sample size re-estimation if you can logistically do it, which you can't always, but when you can, you probably should. but that's a simple adaptive study. That they can be, as you say, you can have all sorts of complicated things with Bayesian designs and dropping arms, adding arms, changing doses, changing treatments, changing endpoints, all sorts of things, and it can get really complicated. And when it does get really complicated, then you really need your statistician. You might need to do some simulations before you start to figure out what the range of possibilities are, and will this complicated adaptive design actually save you any time or money, or will it just make your design more complicated? Sometimes people want to do adaptive designs because they've heard that it's a trendy thing to do. And not all studies need an adaptive design. Sometimes it can be really helpful. Sometimes actually keep it simple is the way to go. But certainly,
yeah, especially if you know you're going to enroll all of your subjects at the interim.
Yeah, and the
there's no need to do it.
For the early phase exploratory studies, you can do some wild and wacky things, and nobody's going to stop you. If you're doing a phase 3 study and hoping to get your license on the back of that, you probably need to be doing something pretty standard. Or if you're not, then really have a good discussion with the agency first, because they're probably not going to like you doing fantastically innovative designs in your phase 3 study.
And I'm looking over here because I have some of the comments we did in our prep, and I think Elizabeth made this comment, and it's probably my favorite, is adaptive doesn't mean you get to do whatever you want, whenever you want in your protocol. There's still methodology that goes into play around creating the appropriate adaptive design. And speaking of early phase studies, you can still bring a lot of statistical rigor, even in phase 1.
Yeah, I like this one too. This is a great point. Anytime that we are making decisions, we should make sure that those decisions are made based on data that are collected correctly, analyzed correctly, interpreted correctly, and that means statistical rigor. So, we assume if we're doing a clinical trial, it's because there is information we want to gather, that there's truth that we want to uncover. And we have to make sure that we do that right, or else we could get led astray.
Yep. And I think statistical rigor is context dependent. And probably the most important thing here is to draw a distinction between late phase confirmatory studies and early phase exploratory studies. Because in the late phase confirmatory studies, that statistical rigor goes to regulators' risk. The regulator is not going to approve your drug if they think your statistics aren't rigorous enough. And that's kind of important. In the early phase studies, it's really sponsors risk. If you're making decisions on the back of your early phase studies about what to do next and you get it wrong, no patients in the big wide world out there are going to be harmed because your drug's going to fail before you get as far as the license, or if it doesn't, then you're not going to get your license. But you might have then just wasted 50 million quid on a huge phase 3 study that you really shouldn't have done in the first place. So you may have different statistical rules, but at the end of the day, you want to make a good decision, however that's defined.
Please don't ask us for just a quick sample size calculation. I think that's the other button on the keyboard, is just press that button and give me the sample size. Does anybody want to talk about what goes into sample size calculation here?
Sure, I can take that in part, at least. Our sample sizes, the design, like none of this happens in a vacuum. I think this is, a consistent topic that came up throughout all of the items that we're discussing today. Clinical trials are inherently collaborative. I am not a theoretical mathematician. And that's because I like to do math that is grounded in something in the real world that has an impact on actual people. And but that means that when we do a study design or a sample size calculation, what we need are thorough, well-researched, informed ideas from our clinical colleagues and biological or preclinical, as the case may be, because that is what guides the statistical decisions. All statistical decisions should be grounded in biological medical reasoning. There should be a reason that we choose a certain design. There should be a reason that we choose an endpoint. These are not necessarily statistical decisions, but they're the information that all of the statistical decisions are built upon. And if they're built upon poor assumptions, then that can have very wide-reaching and negative impacts for the trial and for the program.
Adam, anything to add?
Well, I think the word assumptions that you just used, Elizabeth, is really key there, because your sample size is not based on facts, it is based on assumptions, and in some cases you may have really good data on which to base those assumptions and you can be pretty confident of them, and in other cases you may really not have good data and it may be just an educated guess. And it's always worth bearing in mind that your sample size calculation is only as good as the assumptions that went into it. And I mentioned earlier that I'm a great fan of sample size re-estimations. If you can check your assumptions when you're halfway through the trial, that's a really useful thing to do. You can't always do it, but when you can, it's a great thing to do.
Yeah, and I often find myself cautioning clients who they have assumptions for their sample size calculations, and then they get their p-value at the end and it's successful, but the point estimate wasn't quite what they thought it would be, and so they were thinking that just because I got the p-value, then my point estimate is good as well, or it's what was assumed previously, and I've kind of fumbled through that, but so then I have to explain the concepts of power and sample size. But yes, it's really important to sample size gets them in the end.
And Adam, you mentioned power, and I've got a couple of statements here. We're going to start with this one, which is very high power isn't everything. So one of you please explain power to potentially people who are not statisticians, and then talk about why just because it's high, that isn't everything. Elizabeth?
Yeah, oh, sorry, go ahead.
Or go ahead, Adam. I don't care. Just
yeah, I mean, you make assumptions, you make sample size assumptions based on your treatment estimates, variability, et cetera. Okay, but you're not, that's a single point estimate, and you're not going to exactly hit that, right? So if you want to make sure that you account for all of the different contingencies, for example, okay, then you increase your power. Now, the technical statistical definition of power is, failing to reject the null hypothesis when the alternative is in fact true. And some people think of that as a type 1, type 2 error and such, and you can explain it that way. But it's really, I tell folks it's almost like hedging your bets. Okay, you want to make sure the study is large enough where if odd things happen and you deviate from your estimate somewhat, you can still come to the right conclusions. So.
Adam
or Elizabeth want to add to that?
I would add to that just to say that, obviously, if there is something to be seen, if there's a treatment effect, we want to be able to see it. That's kind of the idea around power. And it's not everything, the desire to make sure a study is extremely highly powered. Industry standard is somewhere between 80 and 90%, depending on, what phase, et cetera. If you're trying to go above that 95%, 98% power, like you're really experiencing diminishing returns at some point. You're having to enroll a lot more people, or, see a significantly bigger effect, in order to see powers that big. It's not everything. I think, in clinical trials, in statistics, there's no one thing that is everything, that is the end all be all. We always have to take every piece of information as part of a whole. And when it comes to power, a big thing to consider is perhaps, what is the size of the treatment effect you're seeing? If it's very, very small, if it's not clinically significant, then having great power isn't really going to do a whole lot of good necessarily. And then you have to consider other things as well. What's the safety profile? What is the market landscape? So high power isn't everything. Always take it in consideration with the whole, with what the drug does, how it works, how safe it is, how much of a treatment effect you see. Everything has to be considered holistically, and not just like, oh, but there's really high power. It's not.
And if we talk about high power, is it correct to say that the higher the power, the more, generally the higher the power, the more we're minimizing the risk of missing the treatment effect? And so the flip side of that would be missing statistical significance of that treatment effect. However, a small p-value isn't everything either, because eventually you go to enough patients, you're likely to find some significance, and is it actually meaningful? And so, Adam J, you want to talk a little bit maybe about why a small p-value isn't the end goal?
So obviously, for regulated clinical trials, if we want to get our drug license, we need to have a p-value of less than 0.05 in our confirmatory trial, or the regulators aren't going to be interested. But that's not everything. I mean, as my colleagues have said earlier, you also have to consider the size of the treatment effect. And if you've got this amazing new analgesic drug that reduces your pain by 1 millimeter on a 0 to 100 millimeter visual analog scale, like, who cares? And if you had a large enough study, maybe that would be statistically significant, but it's not meaningful. I think I'd just like to take that a bit wider and say p-values per se are not everything either. P-values are very widely misunderstood. People often think this is the probability that your null hypothesis is true, but it's not. But sometimes it's not a very intuitive and useful way of thinking about things. And I would like to see Bayesian statistics used more often in early phase trials. I don't think it's coming to late phase trials anytime soon, but in early phase trials, you often want to know, well, what is the probability that this drug actually works well enough to take it on into phase 3? And that is very much a Bayesian formulation of a question, but we don't see that as often as I think we probably should.
And I have to agree with
Oh, go ahead, Adam.
you on the Bayesians. It's really the I've grown into Bayesian tolerant, I guess, but I do use it in a lot of my early phase, and it is from what I've seen. So.
All right, data. Data means little without subject matter interpretation. Avoid going fishing in your data. Who wants to start?
The dreaded fishing expedition. Yeah, I understand where companies are coming from. We went through all of this process to collect these data. We want to wring every bit of information out of it we can. So go forth and do 800,000 statistical tests, none of them adjusted for multiple comparisons. And let's just see what we find. This can get us into a lot of trouble. If you look often enough, you will find something. Is it medically or biologically meaningful? Probably not. If you're looking across, hundreds or thousands of comparisons, anything that we do, again, once again, you have to take things like as a whole. You have to take, also you need to consider everything from all perspectives. It's not just statistics. We need to be grounded in clinical and medical biological information. Um, just looking around and finding things is perhaps not a great idea. Anything that we do needs to be grounded in biological or medical hypothesis about something about the disease process or the population. We need to have a reason to do what we're doing, because if you go out and you do a gazillion statistical tests, then just by sheer chance, you're going to find something that looks real, some association that looks real. And then how much time and resources are you going to throw down that rabbit hole? So like, let's keep things focused and grounded in real world hypotheses. And I think that's the most efficient way. That is the best way to get your value out of the data. It's not to just go flying willy-nilly into as many different comparisons as you possibly can.
And I think this kind of actually brings.
I have to agree. Yeah.
Sorry, Adam, go ahead.
Well, I'm just saying, I think there's a time and place for everything, and in my opinion, this is one reason why real world evidence has gained a lot of traction in the last few years. That's all. A lot of times that's the ultimate fishing expedition. Now, if you apply the appropriate statistics to it, for example, data matching and such, this is where you can get leverage with external controls and such. And so, I don't mind the fishing trips, but they have to be done with a purpose in mind and be able to back it up statistically. Okay, sorry, go ahead.
So I was going to say, I think this actually ties in nicely to the next point you would, in fact, that you just did bring up on the screen. Primary endpoint means one endpoint. If you have thought about your primary endpoint properly, and you absolutely should do that, and you should put a lot of thought into it, and you should discuss it with everybody, and you should read the literature, and you should make sure you've got a good primary endpoint. If you have thought of that properly, 9 times out of 10, you know whether your drug works or not just by looking at the primary endpoint. You don't need to look at 25 other endpoints as well. Now, if you must look at 1 or 2 secondary endpoints, then sure, go ahead. But you generally know very quickly from your primary and maybe 1 or 2 secondary, does this drug work or not? And if it doesn't work, it doesn't work. There's really no point in then dredging through all your 300 subgroup analyses, because it's not going to be meaningful.
Of course, on the CRO side, that's great because we get paid for all of that. But I think that you have to think about what you're looking for, and did you see that or not? And if you want to look for some evidence of what to pursue next, then some of these digging deeper can help. But you're not going to gain some. It's not likely in a well thought out and designed study you've missed some key component.
Yeah.
But this.
And I think.
Go ahead.
I think certainly in a phase 3 confirmatory trial, that primary endpoint means one endpoint is very good advice, because there you really are trying to get statistical rigor and show convincingly that your drug works. Now, early phase exploratory studies, maybe you have a bit more wiggle room there. Maybe at that stage you maybe don't know what your endpoints are because you're still learning about the drug, and that's fair. But again, you probably still need to think about them carefully and pick a small number of endpoints that you think might be good candidate endpoints for your later phase trials, rather than just throwing the kitchen sink at it and hoping that something sticks. If I'm not mixing my metaphors.
Right. It's okay. So we've got the, obviously you want to have one endpoint, but we've got a couple more points to make about endpoints, I guess. And that's where you have the insistence that you have co-primary endpoints. And that makes me shed a small tear sometimes, because what is a co-primary? Then nothing's primary. But what do you do if you have 2 endpoints that are both equally valid and both need to be showing significance? Thoughts on that?
Yeah, so I mean, I don't have any issue with co-primary endpoints. It's when they come back and say, Yeah, we're not going to adjust for multiplicity. That's when I start to raise my hand and say, that you have to adjust for this because uh, it's a basic statistical tenet, right? I mean, so I don't have any issue with multiple endpoints as long as they are handled correctly. You want to do hierarchical testing on the secondaries, that's fine. What's the stipulation? Well, both of your primary primary endpoints need to pass and such. And so, as long as you approach it from that standpoint, um, then I have no issue with that. But if you don't adjust for multiplicity, the agency will certainly come back at you.
Yeah, when we're talking about adjusting for multiplicity, we're talking about how are we spending that alpha, and how are you, if you had your alpha is the significance level, and we were talking about power earlier, and that's commonly referred to as beta, but how are you spending that alpha and spreading that across your endpoints, and how have you set up the structure of testing so that which one goes first and which one goes second? And I think that drives into another component, and I think, Elizabeth, you have a lot to say about this, especially in context with the first one, is now you have co-primary endpoints, and then you have these key secondary endpoints, and what do all these things mean anyway, and how do they impact how you're interpreting your study?
So I'm going to back up a little bit and say, alpha is directly related to how high do we need to set the bar for success. So, we only have so much alpha. And if we're going to spend all of our alpha on a single primary endpoint, that will make the bar as low as we can get it in terms of the statistical tests that we're going to do. If you have two co-primary endpoints, I feel like sometimes companies have this idea, then if either one of them is good, then we're going to be, then it's a success. No, it's not either. It's both now. You have to have both to be significant. And because you have to share alpha among them, you raise the bar for both. So you're really setting yourself up to, a potentially challenging definition of success. So, but it comes down to, what is the definition of success for your drug? Do you need both of these things for it to be considered successful? Then yes, you should have both of them in there. And so, and this idea sort of pours over into key secondary endpoints. It's, again, what is the definition of success? What does success look like for your drug? And these key secondaries are sort of, these are supporting, these are things that we're hopeful about what the drug does, but they're not the meat and potatoes. The meat and potatoes of what success is our primary or our co-primary endpoints. And then the secondaries are things that we think we might also be able to get, but these are just great to haves, not necessary to haves. So again, and this again comes back to collaboration. We need our medical, clinical, preclinical colleagues to help us define, and sometimes, our perhaps commercial marketing colleagues as well, to help us define what success is. The choice of endpoints is not a purely statistical decision. We will help you figure out how to test those in a statistically rigorous way so that we are, allocating alpha appropriately so that we're controlling the family-wise type 1 error. But this is a decision that comes down to a holistic view of what the drug does, what success looks like.
And I'm going to attempt to remember my statistics from back when I was working every day as a statistician, but just to underline it for people who maybe aren't statisticians in here, and we're talking about spending our alpha, and we talk about a common p-value of less than 0.05. And so we can think of 0.05 as the alpha budget. And how are you going to divvy that up? And that's what we're talking about when we talk about what are the algorithms and the hierarchy for these tests. And if you have two co-primary endpoints, you might say, okay, we need to spend all of our alpha on these two endpoints. And so maybe it's 0.025 for each of them, or maybe there's a reason that you divvied up a different way. And then if you are bringing in key secondary endpoints, you now have to save some alpha for those too, if they're going to directly impact whether you're saying your drug is efficacious or not. I think if you've got a couple of primary endpoints, you're probably looking at both of those, and that's great, and you're good. And the secondaries tend to be just supportive, interesting things that might be helpful for patients as well. So you just have to think about an alpha budget, and it's 0.05, and how are you divvying that up, and what rules are you following, right? Like you can't spend it on your secondary endpoints if your primary endpoints are not significant.
Yeah, and that's normally the way we do it. We wouldn't necessarily split the alpha with the secondary endpoints. We just say we're not going to look at those secondary endpoints unless the primary endpoint is successful. And if you do that, then you can still use your whole 0.05 on the secondary endpoint, but you kind of have to do it in that order. Then you have to say which one are we going to do first, and that all has to be carefully pre-specified.
And I think that I want to underscore that it needs to be carefully pre-specified. That means you need to be thinking about this when you're writing the protocol, and you
Always.
sure need to be thinking about it when you're writing that analysis plan. And the clinicians and the other stakeholders who are reviewing the analysis plan need to be understanding and agreeing that this is the way to go. The statistician will have the technical and theoretical advice on how to do this, but this is where you want to make sure you're making the right decisions that make clinically meaningful interpretation of your data as well.
Right. And there are a lot of different techniques that you can do so that you can start testing with all of your alpha, and then continue if your p-values allow it. Step up and step down methods and such. I won't get too technical with this audience, but there are ways to present it such that all of your statistical rigor is maintained.
Yep. And I think the key there, just to underscore, is the pre-specification. And a lot of statistics comes down to pre-specifying what you're going to do and then following those rules. And you're not changing them at the end just because you didn't quite see what you wanted. We're going to shift a little bit, still related to endpoints, but in a much more pragmatic, practical way. If you're using any kind of scale or instrument or measure for the endpoint, make sure you provide the references for scoring algorithms.
Yes, please.
Yeah.
I'm a statistician. I don't believe in just looking at the numbers. I think as a statistician, part of my responsibility is to understand the context in which the numbers exist. And when it comes to things like surveys, obviously they were designed and built in such a way and validated with a specific scoring algorithm in mind. And not only do I need to know that, like I personally want to read the whole instrument. I want to read the foundational paper. I want to read the validation methods, because because sometimes, senior management or a sponsor company will come to you and say, I want this. I want analysis. I want the average of like this group of questions, or I want like some overall score, or, in order for me to understand if that is meaningful, if that is something that really should and can be done with the survey as it's built, I need to have gone through and make sure that I understand what it's asking, how it's scored, what is meaningful. So I'm a big believer in source documentation for anything that I'm trying to do, but specifically certainly for things like scoring surveys.
I think it's worth drawing the distinction here, if you're looking at rating scales that need a scoring algorithm, between ones that you pay for and open source ones, because some of them are copyrighted and you have to pay a license fee to use them from the copyright holder. And if you're doing that, they should give you a detailed scoring manual. I mean, not give you, because you paid them a lot of money for it, but they should provide you with a detailed scoring manual that should tell you everything you need to know about the scoring algorithm. And if you don't get that, you should be asking for your money back, because that is their job to provide you those details. If it's an open source thing, then it's kind of up to you to track down the literature and hope that gives you enough detail to do it properly.
Yeah, I'm going to make one comment, and I'm going to keep us moving along because we want to get through all 20 things. But I think there's some There's some scoring for instruments that are a black box, and you need to make sure you can access that black box to get your scores done. Sometimes there's different rules for handling missing data, or if only one sub-score is missing, and you need to understand what those rules are. And I just gave 2 examples from things I personally have encountered over the course of my career, and it's left us really scrambling for how are we calculating the score. So as the statistician, your job is to, ideally, as Elizabeth said, understand everything you can about it, but at a minimum, know how this thing is being scored and what rules you have to apply to maintain the integrity of the scoring. Moving on also to data. More data doesn't mean better data. Try and limit data collection to the essentials. Well, I had to try, but do limit data collection.
I mean, I think this ties back in nicely with what we were talking earlier about fishing expeditions. You need to know what data are meaningful to show that your drug works, or whatever you're trying to achieve from your trial. And once you figure that out, stop. If you collect data on another 12 endpoints, you're not going to learn any more from it. You're just going to spend a lot more money and upset all the patients who've got to spend 3 times as long at the clinic visits because they've got to do all these extra scoring instruments, and make your data harder to interpret and make the investigators more at risk of messing things up because they've got so many things to do, they got confused. So yeah, know what you're collecting and why you're collecting it, and just collect what you need.
Oh, Adam, I think you're muted.
You're still muted.
While Adam's trying to get himself unmuted, I will say, one way that I like to think about this is in personal finance, there's like an idea that if you're putting together a budget, every dollar should have a job. And it's the same thing with data collection. Every piece of data that you collect should have a specific job that it is doing. And that means that it should be grounded in, once again, clinical, preclinical, biological hypotheses, and it should be going towards something. We should have an idea of how we're going to use this.
All right, great. Thank you. I'm moving on because here's another one about data. Third-party data is as meaningful as CRF data. So much data is collected outside of the EDC database. So make sure your statistician is seeing those data transfer agreements and can review those test transfers.
Yeah, and sometimes it can be more meaningful than CRF data. I mean, I've worked on trials where you actually get your primary endpoint data from some sort of third-party source. And I would say not only do we need to kind of think about that statistically and make sure we've got the data we need, but just also logistically, because sometimes these non-CRF data can actually be a real pig to get incorporated into your CDISC data sets. And you need to make sure you've got time and budget built into your project plan for that.
Yeah, absolutely.
All right, I'm bringing up one. We thought this was a little spicy that when we were talking about it, because we're saying it very nicely, but there's a lot of real thought and feeling behind it, which is caution and balance are needed when accessing data during the study. There's lots of reasons people are going to be reviewing data during the study. Know why you're doing it.
Yeah, I'll go ahead and take a crack at this one. Once again, clinical trials are very expensive, and for those of us doing them, but in particular senior management and C-suite folks, they feel like, we're spending all this money. I want to know, and there are lots of reasons to want to know. I want to know because I want to make internal business decisions for the company. I want to know because I want to share this information with our investors so we can raise enough money to finish this study. I want to know because We have a formal interim analysis planned that is going to give us an idea, with statistical rigor of where we're at in this clinical trial. And and these are all understandable reasons, but for each of these reasons, we need to be very cautious about lots of things. We need to be cautious about how mature the data are. We need to be cautious about any statistical implications. Any time that you're doing a formal comparison, that you're applying a statistical test, we need to keep in mind that adjustment for multiplicity, that idea that the more we test, the more we'll see something just by random chance and not because it's a real effect. We need to keep in mind ethics, especially for a company that's publicly traded. We definitely want to stay far away from the radioactive idea that any insider trading is happening. But in a more broad sense, when data are immature, and if those data are used to make decisions, those decisions might have to be walked back. No one wants to walk back a decision. So, listen to your statistician about what can be done with the data, whether the data are ready to be seen. Listen to legal, listen to your regulator, listen to, all of your key stakeholders as to, what this process should look like and what can be done with it, because you can get yourself into a lot of trouble when you access those early data, depending on what you do with it and how it's done.
Yeah, I'm testing my mic out. I apologize.
You're back.
If,
we can hear you.
can you hear me now? Oh, okay.
Yep.
Yeah, my headset mic decided to stop working all of a sudden, so I had to change it out. I've heard a lot of what you said, especially the issue of accessing data during the study. Now, with different EDC systems and different client access permissions, they can certainly access the data, but I agree that no unblinded data should ever need to be accessed during a study. And if it does, for an interim analysis and such, it's all very well documented and audited, especially if you're using the right transfer methods. So I'll stop there.
I myself was just muted. Statisticians can program, but a dedicated statistical programming team is the better option. And I'm laughing because way back in the day when I was a statistician, I did my own programming. And, now we have these wonderful statistical programming teams, and they're a much better way to get that work done and provide a better traceability in QC.
So statisticians can and should program, in my view. They shouldn't necessarily do a lot of programming because, we hopefully have a dedicated statistical programming team, and they're going to do a lot of things more efficiently than the statistician can. But actually, if I'm working as a lead statistician, I always like to do at least a little bit of programming, maybe my analysis data sets, because then I just feel like I'm getting to know the data a bit better. If I'm actually creating a data set and programming it, maybe not all the data sets, but some of the key ones, then I know exactly what's going on in my study. And I think that's really helpful. But yeah, a lot of the thing, doing our SDTM data sets, our listings, that's not an efficient use of my time. We have programmers who will do that much more quickly than I could do it.
Yeah, and I think if I had one piece of advice I would give to today's generation of statisticians is get your hands dirty in the data. It is so easy to not, because you have teams doing SDTM and doing atom programming, but nothing beats knowing what's going on in that raw data or programming those analysis data sets yourself, because you are responsible for what.
And I think that goes back to the scoring algorithm, right? It goes back, if something goes odd in your data at the end, and you're in a pivotal trial, for example, who's the client going to go to? The lead statistician or the programmer? So they're going to come to the lead statistician, and they will expect you to know what, the algorithms and such, the derivations, and be able to defend those if you need to. So
All right. I asked this in everyone. It's kind of not a 20 thing, but every panel's had it. A quick one statement on how you think AI will impact your job in statistics.
I think it may change some things. AI is a tool, potentially, like any other tool. But, clinical trials are inherently collaborative, as we've discussed. They inherently involve multiple people, and humans need to talk to each other and come to consensus. And we each have our own areas of expertise. And things work best when each of us brings what we can to the table, and all of these things are brought together, and we collaborate in such a way to build a great trial. So I know that there are people who are concerned that AI is going to eliminate the biostatistician in clinical trials. I really don't see that as happening. I think maybe it'll just be another form of programming one day. Right now, obviously, it's still in its pretty nascent state, but who knows what the future holds. But I don't see it eliminating biostatisticians.
Well, I do see it eliminating biostatisticians, but not for the reasons you think. AI has an enormous carbon footprint, and the planet is basically burning, and we are completely failed to get on top of that. And if we carry on throwing around carbon-intensive technologies like AI without thinking about it, there's not going to be any clinical trials in 50 years' time, because we'll all be living in a Mad Max wasteland. And then what are biostatisticians going to do? Which is not to say, AI doesn't have some uses, but I think we do need to be careful about it. Honestly, I don't think there's any part of my job that could be done by an AI right now. Maybe in 10 years' time, that will be different. I have heard some good things from medical writers about some of the more routine parts of that, like serious adverse event narratives. Yeah, maybe an AI can help with that, but I think we need to be careful and only use it if it's really got proven value.
Oh, I couldn't agree more. I mean, is AI going to have the ability to talk through negative results with clients in a manner that they can understand and you can empathize with them? Probably not. Can you utilize AI to automate common SDTM programming? Yes, and I've seen it happen. So there are a lot of things about AI that are great, and certainly if you go to any conference, the vendors I've noticed they're almost 80% AI companies now, and so we need to understand the technology certainly if we're going to keep up in the clinical world, but replacing statisticians completely, I don't see it. And maybe I'm just being a little selfish myself, but I'd like to think not.
Well, I always say, they're doing clinical studies on humans means you get very one-off, interesting data issues all the time. And I don't think that that's something that AI can just magically, where it's at right now, account for. I think it's human in the loop. And I always say, you won't lose your job to AI. You will lose your job to someone who is using AI in a way that makes them more efficient. We're going to speed through the next few so we can get through them all. This is really advice for sponsor side, I think. So I'm going to put both of them up there because we can talk about them at the same time. But one, make sure your statistician is qualified. If you are getting a lead statistician, make sure that they know how to be a lead statistician. And I will let the panel talk about what that actually means. And then the second is know your CRO, know how they are working, and stay engaged with what's going on with them.
Yeah, I'll take a crack at this first. Yeah, here's the thing about being a biostatistician working in pharma is that I would say there are very few, if not zero, doctoral or master's programs that really prepare you for this field. There's so much knowledge involved. Everything from, regulatory, understanding ICH, understanding just the workflow. What is a dry run? Like the practice of writing a statistical analysis plan. These are things that are very, very rarely touched on in degree programs. And so in order to know that your statistician can do their job, it's really key that they have experience under their belt, that they've been involved in study startups, in study submissions, and regulatory conversations, that they've designed studies, that they have the experience in the phase of trial and perhaps therapeutic area that you're functioning in. There's a lot to know. There's a lot that goes into our jobs. And then as to the second point, I know CROs are so important. I mean, our industry is basically built on CROs. They are a vital organ of the pharmaceutical industry. And I know that a lot of companies are functioning on very small startup budgets, and they're really depending on the CROs to take charge. But I have seen the set it and forget it model not pan out. And, the truth is that quality of work can vary widely, even among the biggest CROs. So I think that it's key to ask tough questions and to, again, be sure that whatever statistical lead you're being given knows what they're doing and has a good rapport with their team, and that the entire team has at least a minimum level of experience, and to understand the organizational structure, who has responsibility and accountability at the end of the day for those deliverables. And if all of this is, overwhelming for a sponsor, hire a consultant. There are those of us out there who can help, just stepping in for a few hours a week, even to just, make sure that your statistician does have the experience they need and knows what to look for and can keep things, on time and on budget. So there are good ways of doing this.
And I would add that, having a good statistician obviously is important, but it's not just about the statistician. It's also about the support network around them, because, I'm sure every sponsor would like to have that principal statistician with 20 years experience and 3 PhDs working as the lead on that project. Not going to happen. She's busy with someone else's project. Sorry. But if the statistician who is working on your project, maybe they're not all that experienced, but kind of just experienced enough to be a lead, if they've got those senior colleagues around them who can support them, then that's going to work a lot better than if that person who's only just ready to be a lead is just told to go away and get on with it.
Yeah, and I think you need to recognize, and I think clients also need to recognize, statisticians out of school, they have to start somewhere. So you're training them, but just like in a training hospital, there is someone that's accountable. Okay. And I think it's important to note that, you also, as a manager, you need to recognize when someone is, I call them special, someone gets it, the engagement with the client and such. Certainly you can't really teach that. You can't teach relationship building. You can't teach the ability to speak to a client as if they are your colleague, not necessarily a friend, but they want to know what you know. And so it's very important to recognize that, I don't want clients to get hung up on, well, this person only has 2 years of experience. Yes, but I think if you meet him or her, you'll understand why I'm putting this person out in front of you. So it's kind of it's personal for me because I've had so many wonderful statisticians.
Yeah, and I'm just going to add, and this is the whole thread I could do a whole nother discussion on. But if you are a higher level statistician and you've been working for a lot of years and you're listening to this conversation, take your junior statistician with you to a meeting and let them listen. If you're having a session talking about your regulatory strategy, find somebody that you're mentoring and take them with you so that they can listen. Because will they know what's going on the first conversation? Maybe not, but that is how you build your experience bank, by hearing and being exposed to these items. And we don't get that as frequently as we used to when we all worked in person in offices. And so, try and reach out. And if you are that newer statistician, ask a more experienced person on your team if you can tag along to a meeting. I know that's easier said than done, especially in CROs where you really have to watch your time, but start showing the interest so that you can start getting the exposure. We are at item 20, and I'm just going to say it so I can give these guys an opportunity to say the last word, because I know we're just at time. But just like every other function, make sure you are including your statistician on project updates, timeline shifts, and milestone requirements. They will do everything they can to support you, but they have to know what is going on so that they can plan appropriately. So they're a vital part of the team, and include them that way. Okay, last word from each of the panelists about what you really want people to know about working with their statistician.
So I'm just going to repeat what I said at the beginning, that, a statistician is one part of the team. It is all about teamwork. Talk to your statistician, involve the medics, the project managers, everybody else. Make sure everybody is working well together, learning from each other, respecting each other's expertise, contributing based on their own expertise, and discussing anything that needs discussing in an open and collaborative manner.
Adam Ham?
Yes, I think it's just from the beginning, you need to have the relationship where your statistician, can tell you that you're doing something that is not right, or you're doing something that is not going to pass regulatory scrutiny. I like to tell people, I don't mind doing something crazy, but I just don't want to do something stupid. And that's, I think stupid is where it falls. The regulatory will never pass this. Okay. But I think it's very important for them to be involved from the beginning, build the relationship to the point where you can have professional arguments that end with the right conclusions, with the right decisions. And you won't get that unless you build that relationship early and trust your statistician. So,
Elizabeth.
Wow. Well, everything that Adam and Adam said. To add to that, I would just kind of repeat, it's all about collaboration, and all of the rules of collaboration apply when you're working on a cross-functional clinical trial team. So that means that each person should be trusted with their area of expertise. They should have responsibility and accountability for everything within their function. And as statisticians, from our colleagues, we need those real world, that clinical knowledge, that medical knowledge that we don't have. We need that from you, and from you, we also need trust. That sometimes I think biostatisticians are kind of seen as a little bit of the bad guy, a little bit of the buzzkill, because we say things like, "You can't do that," like, "This design doesn't make sense," "These data were not collected correctly." You can't do that. And just to understand that where we're coming from is the desire to do the best that we can to honor the people whose time and experiences went into these data, so that these data can be used appropriately in a way that will actually reveal as close as we can get to the truth.
I love that, because at the end of the day, everything we do is for those patients. So let's make decisions that are respecting them. I just want to say thank you to everyone who's watched, and to Elizabeth, Adam, and Adam for bringing their experience and their expertise to this discussion. I've loved it. I've enjoyed it. You guys have had great points. If anybody out there has a topic or idea that they would like us to talk about, drop me a line and let me know. Thank you all, and have a great rest of your day.
Thanks, everyone.
Thanks. Bye-bye.
Thanks, everyone.
Thanks.